on.
Hi and a very good morning to all my friends and also lecturers. This is our second video and our second assignment. It is all about enzyme. My name is Ilyas Hanafi bin Razali. My metric number is 182260.
My name is Ui 184874.
My name is Lau Kasim, metric number 182531.
My name is Krishna Veni, metric number 184970.
My name is Zakirallah binti Zakaria, metric number 181839.
And my name is Nur Aina binti Adnan. My metric number is 181826.
So guys, are you ready for this discussion?
Yes.
So we will start with Oizy.
Okay, I will start with. Do you guys know why are enzymes important in the body?
No.
Let me tell you. Many chemical reactions required for the living cell do not proceed fast enough at the pH temperature of the body without enzymes, which means that many processes cannot be done without the existence of enzymes. And what is enzyme? Enzyme is a catalyst, or so-called a specialized protein that catalyzes chemical reaction by increasing rate of velocity or conversion of a specific substrate to a specific product without itself being changed in the overall process. The protein component of some enzymes require other additional components in order to make the enzyme function. Enzymes require additional components known as cofactor for their catalytic activities. Cofactor can either be metal ion, for example like iron ion, magnesium ion, and etc. And organic molecules that often derive from vitamins that known as coenzymes. Not all enzymes require cofactors, but only some does. The complete catalytic function enzymes contain protein and cofactors, which is holoenzymes. That's the basic characteristics of enzymes. And
Here I will add some things on characteristics of the enzyme. Enzyme is often end with "ase", such as enzyme cyclase, lipase, maltase, and it is usually high specific and only produce expected product from numerator or substrate. And it is usually function in moderate temperature or pH, but when enzyme is exposed to extreme pH or temperature, it will become denatured. And another thing is enzyme is neither consumed or nor produced, but during the course of the reactions. But it may become temporarily convertible to molecule that been transformed in the intermediate stage of the reaction. But at the end of the reaction, it will the enzyme will again be in the original forms and as the product released. And next is the enzyme do not take place the reaction, but they enhance greatly in the rate of reaction that would much proceed slower in their absence as a catalyst. And lastly, enzyme may alter the rate of reaction but do not change the amount of product produced. That's it. That's about characteristics of enzyme.
Thank you, Ali. So, do you all know what is the general concept of enzyme?
no.
So I will explain to you. For enzyme to function as catalyst, it must first bind to enzyme, and the second one, lower the energy of activation, which is in transition state, so that the reaction will occur at a faster rate. So the first one, binding a substrate to an enzyme. We have two model, which is log and key model, and also induced fit model. How does log and key model function? In the log and key model, the enzyme is assumed to be log and the substrate is the key. Only one part of the substrate will fit into the active site of an enzyme, are made to fit exactly to allow the reaction to occur. So the substrate are made to fit exactly with the enzyme. This model fails to take into account the fact that proteins can change their conformations to accommodate a substrate molecule. So enzyme can also change their conformation model to fit with substrate molecule. So thus, the second one is induced fit model. The induced fit model of enzyme action assumes that the enzyme is flexible, and after the substrate bind to the enzyme, the conformation of protein changes to accommodate the substrate molecule to form a stable enzyme substrate complex. After we bind the enzyme with substrate, we must make sure that the transition state happen. So the transition state is conversion of substrate to product proceeds through the formation of transition state. The free energy of uncatalyzed reaction is very high. Enzyme from favorable interaction with the substrate facilitate formation of the transition state by lowering the free energy of activation. The transition state then dissociates to give the product and regenerates free enzyme. So before the product is produced, it will go through transition state, which has lower free energy. I think that is all the general concept of enzyme mechanism. Can we know the classification of enzyme?
Okay, sure. So now I will talk about the classification of enzymes. Basically, enzymes can be classified into 6 classes, which are the transferases, oxidoreductases, hydrolases, isomerases, ligases, and lyases. Oxidoreductases catalyze the reaction of transferring the hydrogen and oxygen atoms or electrons from one substrate to another. The examples of oxidoreductases are dehydrogenases and oxidases. And next, transferases will react by transfer the specific group, be it a phosphate group or a methyl group, from one substrate to another. For example, transaminase and kinases. Whereas hydrolases will catalyze the hydrolysis of a substrate. The examples of hydrolases are esterases and digestive enzymes. And next, isomerases will react by changing the molecular form of the substrate. For example, phosphohexoisomerase and fumarase. Lyases will react by remove a group or addition of a group to a substrate. For example, the carboxylases and aldolases. And citric acid synthetase is the example of ligases, which react by joining 2 molecules by the formation of new bonds. These are the classification of the enzymes.
Next, I will further brief about enzyme inhibition. Okay, do you guys all know what is an enzyme inhibitor?
No.
Okay, enzyme inhibitor is a compound that binds to an enzyme and interferes with its activity, which means it will block the formation of the product. So when inhibitor binds to the enzyme, it will prevent the formation of enzyme substrate complex first, and then the inhibitor will, as bind to the enzyme substrate complex, it will block the chemical reaction to take place, which leads to the formation of product. But when the inhibitor binds, no products will be formed. So type of inhibitor, it can be classified into 2. First is reversible inhibitor and non-reversible inhibitor. In reversible inhibitor, we have competitive, non-competitive, and uncompetitive. So for competitive inhibitor, the structure of the inhibitor resembles the structure of the substrate, which means the inhibitor will bind to the active site of the enzyme while competing with the substrate. So it will only bind to the free enzyme. So increasing the concentration of the substrate can usually overcome the inhibition. If we see the competitive inhibitor, it does not bind to the same site of the as the substrate, which is the active site. It will bind to other side of the enzyme, which alter the shape of the enzyme and prevent the formation of the product. Next, for the non-competitive inhibitor, even though we increase the concentration of the substrate, the inhibition cannot be overcome. If we see the uncompetitive inhibitor will bind to the enzyme substrate complex, so no reaction will take place or no products will be formed. So the inhibitor will bind to the enzyme substrate complex only, and increasing the substrate favors the inhibition by the inhibitor. For irreversible enzyme inhibition, reactive compound will combine covalently with enzyme molecule, and it will modify and inactivates the enzyme. So all irreversible inhibitors are usually toxic substance and either natural or synthetic. That's all for type of inhibitor. So Zakira, can you explain further about the uses of enzymes?
Sure. So this is my turn. Oh, sorry. Thank you to Krishna for a good explanation on inhibitor and competitor. So this is my turn to explain to you guys on uses of enzyme in our industrial. Actually, this has 2 types, as a enzyme inhibitor, as a therapeutic agent, and also in clinical diagnosis. So in a therapeutic agent, dihydrofolate reductase is also used in cancer, mostly in cancer. So in the clinical diagnosis, enzyme may be isolated from the same or different tissues. So analysis has shown that it is distribution of enzyme of a particular enzyme is sometimes useful tools in clinical diagnosis. Enzyme mostly used in clinical diagnosis are also present in serum. You know serum?
Yes.
It's my job, not my job. My responsible also to tell you, like serum is like aldolase, lactic dehydrogenase, creatine kinase, also used in clinical diagnosis. And the last one is elevation and depression of the activity levels of specific enzyme may indicate the presence of disease or maybe damage the specific tissue. So, for example, to confer the heart attack, lactate dehydrogenase is an indicator to know that the level of heart attack in someone. Okay. That's all from me on about uses of enzyme. Maybe you want to add something.
So there are other enzymes also that can be used in clinical diagnosis. For example, the acid enzyme. I give 3 examples of acid enzyme. Acid phosphatase, which is used to diagnose prostate cancer. Amylase to diagnose acute pancreatitis, and also creatine kinase to diagnose muscle disorders and heart attack. Anyone has any addition?
No.
Okay. So I think that's all from us for our second assignment on enzyme. My name is Ilyas Hanafi.
My name is Niche.
My name is Lakasin. My name is Krishna Veni.
My name is Akila Manis.
My name is Ainadila.
That's all from us. Thank you. Goodbye.
Bye.